Take a slow, deep breath. Feel the air move in through your nose, down your throat, and into your lungs. Now, let it go gently, easily. As you settle into stillness, imagine the world within your own body beginning to come into focus. Beneath your skin lies a quiet universe, vast, complex, and alive. It is not made of stars or galaxies, but of cells. Trillions of them. Each one a tiny, self-contained world, working in silence. Every heartbeat you feel, every breath you take, every thought drifting across your mind, all are powered by these microscopic structures. They are the foundation of you. Invisible to the naked eye, yet essential to every moment of life. Imagine zooming inward past the warmth of the skin, through layers of tissue, until the body's smallest forms come into view. You find yourself in a landscape of motion and light. Here, the boundaries between living and non-living blur into delicate membranes and flowing currents. Each cell carries within it the full instructions for life, a genetic code written in the language of DNA. Inside every cell, countless molecules move with purpose. Tiny engines generate energy. Thin strands of protein give shape and strength. Chemical messengers pass through fluid pathways, keeping the body's trillions of cells in perfect coordination. Even now, as you rest, this microscopic world is alive with activity. Cells absorb oxygen, exchange nutrients, and repair what has been worn down. Old cells quietly make way for new ones. In every heartbeat, millions of them live, work, and are replaced. Each cell follows patterns set in motion long before you were born, cycles of energy and renewal that have continued through billions of years of evolution. From the first single-celled organisms in Earth's ancient oceans to the intricate human body you inhabit now, this quiet continuity has never ceased. And yet, though they are many, your cells are not separate from you. They are you. Together they form your skin, your blood, your heart, your brain. Each cell plays its part in the great collective that is your living self. When you think, the signal travels through long nerve cells. When you breathe, muscle cells contract in rhythm. When you heal, stem cells awaken to create new tissue. Every moment of your life is a reflection of their harmony. So as you listen and drift further into rest, imagine this universe within you, vast, silent, and endlessly intricate. A cosmos of life, existing not in the sky, but in every corner of your being. This is the world of the cell, the quiet universe within. The world before the microscope. For most of human history, the tiny structures that make up life were invisible. People could see plants, animals, and the stars above, but not the worlds beneath the surface of the skin or inside a drop of water. The human eye, even at its sharpest, could not see beyond a grain of sand. In the 17th century, that began to change. Across Europe, glassmakers were experimenting with lenses. They polished, curved, and stacked them, discovering that they could bend light in ways that magnified what was small. At first, these lenses were toys for curiosity, spectacles, magnifying glasses, small instruments to inspect cloth or insects. But in a few hands, they became tools of revelation. Robert Hooke and the birth of the word cell. In the year 1665, an English scientist named Robert Hooke sat at his desk with one of the earliest compound microscopes. It was not a simple device, a long brass tube, a flickering candle for light, and a mirror to direct the beam through the specimen. The image shimmered and wavered, but what Hooke saw would alter science forever. He examined a thin slice of cork, taken from the bark of a tree. Through his lens, the surface appeared to be made up of countless tiny boxes, repeating chambers, neatly arranged. They reminded him of the small rooms, or cellulae, where monks lived. And so, he named them cells. Hooke recorded what he saw in a book titled Micrographia. It was filled with detailed sketches of fleas, fibers, and plant tissues, objects magnified dozens of times. Readers of the time were astonished. For the first time, the hidden geometry of living matter was visible. What Hooke did not know was that the cork was dead tissue. The cells he saw were empty walls, the structural remnants of plant life long gone. Still, the name remained, and with it, a new way of thinking about the nature of life. Antony van Leeuwenhoek and the Living World. While Hooke worked with a compound microscope, a Dutch tradesman named Antony van Leeuwenhoek built his own instrument, simple microscopes, but with lenses of extraordinary quality. His technique for grinding glass was unmatched, allowing him to reach magnifications far beyond his peers. Leeuwenhoek was not a formally trained scientist. He sold cloth and managed a store in Delft. But his curiosity drove him to explore the unseen. He placed drops of pond water, blood, and saliva beneath his lens, and there, he saw life itself. Tiny creatures darted and spiraled through the water. They were bacteria, protozoa, and single-celled organisms, though the names would come much later. He called them animalcules, meaning little animals. Leeuwenhoek sent careful letters describing his observations to the Royal Society in London. He drew what he saw and counted the shapes and movements of these invisible beings. The report seemed unbelievable, a hidden world thriving in every puddle and mouthful of water, but others confirmed his work. For the first time, humanity knew that life existed at a scale far smaller than the eye could see. The slow growth of understanding. After Leeuwenhoek, microscopes became tools of both wonder and study. Through the 18th century, scientists improved the lenses, but their understanding of what they were seeing grew slowly. Some still believed in spontaneous generation, the idea that life could arise from non-living matter. Maggots from meat, microbes from broth. Simple observations seemed to confirm it. It would take time and better microscopes to reveal that even the smallest forms of life came from others like themselves. Cells, too, reproduced. They did not simply appear, they divided, and from one came two. By the early 19th century, a new generation of naturalists began to look deeper into tissues, not just plants, but animals as well. They stained specimens, sliced them thin, and examined the repeating patterns inside. Schleiden and Schwann, the birth of cell theory. In 1838, the German botanist Matthias Schleiden studied plant tissues under a microscope. He noticed that every part of the plant, from root to leaf, was made up of cells. A year later, his colleague, Theodor Schwann, extended this idea to animals. Together they proposed something revolutionary: that all living things, both plant and animal, are composed of cells. This became the foundation of what we now call cell theory, one of the unifying principles of biology. The theory had three core statements. All living organisms are made of one or more cells. The cell is the basic unit of structure and function in all living things. All cells arise only from pre-existing cells. These simple ideas transformed science. No longer was life an indivisible mystery. It was built, piece by piece, from units so small that only a microscope could reveal them. By the late 1700s, microscopes are common in laboratories across Europe. Naturalists and physicians begin to explore the tissues of plants and animals in greater detail. They find patterns, repeating shapes, repeating structures. But the meaning of those patterns remains elusive. Were these tiny chambers simply the building blocks of larger tissues? Or were they something more, perhaps the very seat of life itself? The answer will emerge in the 19th century, when two German scientists, working separately, make a profound connection. Schleiden and Schwann, the birth of cell theory. Matthias Schleiden, a botanist, studies plants under the microscope. He notices that every plant tissue he examines, roots, stems, leaves, is composed of similar cellular structures. At the same time, Theodor Schwann, an anatomist, studies animal tissues and finds the same repeating pattern. Though their fields are different, their conclusions converge. In 1838 and 1839, Schleiden and Schwann propose what they call cell theory, a unified principle that defines all living things. Its claims are simple, yet revolutionary. All living organisms are made of one or more cells. The cell is the basic unit of structure and function in all living beings. For the first time, the living world in all its diversity is understood as variations on a single theme. Plants and animals, once thought fundamentally different, are built on the same foundation. The same microscopic architecture supports every leaf, every feather, every beating heart. This realization transforms biology. It gives scientists a new way to think about life. Not as a mystery, but as a system built from repeating, self-contained units. Rudolf Virchow, the continuity of life. One question remains. Where do cells come from? Many still believe in spontaneous generation, the idea that life can arise from non-living matter. But in 1855, another German scientist, Rudolf Virchow, adds the final piece to the puzzle. He states a simple but profound idea. Omnicellular e cellular. Every cell arises from another cell. It is the missing link. Life, Virchow explains, does not appear out of nowhere. It continues. Each new cell born from the division of an existing one. From this idea comes a new understanding of growth, of healing, and of heredity. Every living being, every structure in the body, is part of an unbroken chain stretching back through time. The discovery of the cell, and the realization of its continuity, mark one of the greatest revolutions in science. Every discovery begins with a question. After centuries of wonder and decades of study, scientists had finally uncovered the basic truth that all living things are made of cells. But this revelation, simple and elegant as it was, raised a deeper mystery. What is a cell? What defines it as living? And how does something so small give rise to the vast diversity of life we see around us? The cell is the unit of life. The cell is the smallest structure capable of carrying out all the functions we associate with life. It can take in energy from its surroundings. It can grow and reproduce. It can respond to changes in its environment. And, perhaps most remarkably, it can maintain the delicate balance necessary to stay alive. Every plant, every animal, every bacterium is built from one or more of these remarkable units. Some organisms consist of a single cell, a bacterium swimming through a drop of water, a yeast cell budding on the surface of fruit. Others, like humans, are vast collectives of trillions of cells, working together in intricate coordination. Whether alone or in a colony, each cell operates as a tiny, self-contained world, a living system enclosed within a fragile boundary, a world within a boundary. At the heart of every cell lies its defining feature: the cell membrane, a thin, flexible barrier made of lipids and proteins. Though it is only a few nanometers thick, the membrane is essential. It separates the interior of the cell from the world outside, creating a controlled environment in which life can unfold. The membrane is selective, allowing certain molecules to pass through, while keeping others out. It maintains a careful balance of water, salts, and nutrients, and communicates constantly with neighboring cells. Within this barrier, the cell is both protected and connected, a single living unit in conversation with the wider world. Under the microscope, cells reveal a stunning variety of forms. Some are nearly perfect spheres. Others are long, thin, or intricately branched. Some move freely, while others remain fixed in place. Their shapes are not random. They are sculpted by purpose. A neuron, for example, stretches in delicate threads across great distances, carrying electrical signals through the body. A red blood cell, small and concave, bends easily through narrow capillaries, delivering oxygen to every corner of the tissues. Skin cells form tight, overlapping layers, creating a barrier that shields the body from harm. Each form is an adaptation, a reflection of the task that cell must perform. Yet beneath their differences, every cell shares the same fundamental architecture: the inner world of the cell. Inside the membrane, the cell is filled with a semifluid substance known as cytoplasm, a clear, gel-like material that holds all the components of life in suspension. Floating within this landscape are tiny structures, each performing a specific function. These are the organelles, the machinery of the cell. Some organelles build proteins. Others process nutrients or store materials. Still others generate the energy that drives the cell's many reactions. Together, they form a complex, dynamic system, a microcosm of cooperation. If one could shrink down to the scale of a molecule and drift through the cytoplasm, it would feel like moving through a bustling city. Everywhere, molecules shuttle cargo from place to place. Signals are exchanged. Energy flows. And information is copied, translated, and acted upon. There is no stillness here. Only organized motion. A rhythm as constant as the beat of a heart. The nucleus, the command center. In most cells, one structure stands out above the rest. The nucleus, large and spherical, enclosed by its own double membrane. The nucleus is the repository of genetic information. Inside it lies DNA, the molecule that carries the instructions for building and maintaining the organism. Every function of the cell, from growth to repair, is guided by this code. The nucleus doesn't act alone. Within it, a dense region called the nucleus produces tiny particles known as ribosomes, the machines that assemble proteins. Through the pores in the nuclear envelope, molecules move in and out, carrying messages, materials, and signals. The nucleus is both archive and command center, a library of life, carefully protected and constantly in use. Not all cells are the same. Across the tree of life, we find two great categories: prokaryotic and eukaryotic cells. Prokaryotic cells, such as bacteria, are small and simple. They lack a true nucleus, and their DNA floats freely in the cytoplasm. Yet their simplicity belies their success. They are ancient, the earliest forms of life to appear on Earth, over 3.5 billion years ago. Eukaryotic cells, by contrast, are larger and more complex. They contain many organelles, including a defined nucleus to house their DNA. Plants, animals, fungi, and protists all belong to this eukaryotic world. The difference between the two lies not only in structure, but in the story of evolution itself. At some point in the distant past, it is believed that one simple cell engulfed another, forming a partnership that would eventually lead to complex life. This ancient collaboration gave rise to the mitochondria, the powerhouses within our own cells, and it set the stage for the diversity we see today. The scale of the microscopic, the scale of the cell world is difficult to imagine. A typical human cell measures about 10 to 30 micrometers across. That means you could fit roughly 1,000 of them along the width of a single grain of sand. A red blood cell is smaller still, just 8 micrometers wide. And yet, within that minuscule space, exists an entire living system. Membranes, proteins, DNA, energy factories, and transport networks, all operating in perfect harmony. Some cells are much larger. An unfertilized human egg, for example, is about the size of the tip of a pin, large enough to see with the naked eye. Others, like neurons, can stretch to remarkable lengths, sometimes spanning the entire body of an animal. From the smallest bacterium to the longest nerve cell, the diversity of cellular form is a testament to life's adaptability. The cooperation of cells in multicellular organisms. Cells rarely live alone. Instead, they organize into groups, working together to form tissues. Tissues that form organs, and organs that form systems. In the human body, there are roughly 200 distinct types of cells, each specialized for a different purpose. Muscle cells contract, generating motion. Nerve cells transmit information. Epithelial cells line surfaces, creating boundaries. Immune cells patrol the body, defending against invaders. Though their tasks differ, their survival depends on communication. Cells send and receive chemical messages constantly, adjusting their behavior in response to the needs of the whole organism. This cooperation allows complex life to exist, a symphony of cellular activity, with each cell playing its part in the grand composition of life. The balance of order and chaos. Inside the cell, the line between order and chaos is razor thin. At every moment, thousands of reactions take place. Molecules joining, breaking apart, transforming. These reactions are precisely coordinated. In the vastness of the known universe, we search for meaning among stars and galaxies, for patterns in the immensity of space. But there is another universe, smaller, hidden, yet no less magnificent. It lies within us, and within every living thing. The universe of the cell. To truly understand life, we must learn to think in new scales, to look not outward to the infinite, but inward to the infinitesimal, a hidden world of trillions. The human body is a collection of roughly 37 trillion cells. It is a number so large that the mind struggles to comprehend it. If you were to count one cell every second, without stopping, it would take you more than 1,000,000 years. To finish the task, every second of your life, millions of cells die, and millions more are created. Your skin renews itself every few weeks. Your stomach lining every few days. Even your bones, which seem solid and unchanging, are constantly remade. Cell by cell, atom by atom. The body is not a static structure, but a living process, a flowing exchange of matter and energy. You are, quite literally, a river of cells. The scale of the microscopic. To grasp the true scale of a cell, we must shrink ourselves down, imagining a world far smaller than we can see. A typical human cell measures between 10 and 30 micrometers across. A micrometer is 1/1,000,000th of a meter. A distance so small that a single human hair is about 3,000 times wider. If an average cell were the size of a small house, a human body would stretch for thousands of kilometers. We are vast architectures, built from living bricks too small for the naked eye to perceive. And yet, within that minuscule space, there exists complexity beyond measure. A cell's interior is a bustling world of structure and motion. Membranes folded into chambers, molecules forming highways and machines, information flowing in endless cycles. To observe it under a microscope is to glimpse a living cosmos. Not all cells are equal in size. Some are large enough to see without assistance. Others are so small that they defy the limits of modern microscopes. The human egg cell, for example, is one of the largest in the body, about 100 micrometers in diameter. You could just barely see it, as a faint speck on a slide of glass. At the other extreme, a bacterium might measure less than 1 micrometer across, 1,000 times smaller. And even among bacteria, there is astonishing variation. Some live in soil or seawater. Others thrive in boiling springs. or the icy depths of the Antarctic. Each one, no matter how simple it seems, is a masterpiece of molecular design. Among the largest single cells known to science is the ostrich egg, which can weigh more than a kilogram. It is one cell, a reminder that scale in biology is bound only by function and form. The diversity of cells. Cells come in a bewildering variety of shapes and types. In the human body alone, scientists estimate there are over 200 distinct kinds. Each one carries the same genetic blueprint, yet each expresses it differently. A variation on a single theme. Consider the red blood cell, shaped like a shallow disc. It contains no nucleus, leaving more room for the molecule hemoglobin, which binds oxygen and carries it to every tissue. A neuron, on the other hand, stretches across remarkable distances. In some cases, a single neuron may extend more than a meter in length, a delicate thread connecting brain to muscle. Then there are muscle cells, packed with fibers that contract and release in rhythmic motion. Epithelial cells form tight, protective layers, the body's living armor. Photoreceptor cells in the retina capture light and translate it into the language of the brain. Each type of cell serves a unique role, yet all follow the same basic principles. Membrane, cytoplasm, genetic code. In their diversity lies unity, the architecture of multicellularity. In simple organisms, a single cell can live independently, feeding, reproducing, adapting. But as life evolved, cells began to specialize and cooperate. Groups of cells formed tissues. Tissues formed organs. And organs formed organ systems. The digestive tract, the lungs, the brain, the heart. This cooperation is what makes complex life possible. No single cell in your body could survive alone. A neuron cannot digest food. A stomach cell cannot send a thought. But together, their abilities combine into something far greater than the sum of their parts. This is the miracle of multicellularity. The transition from independence to interdependence. Somewhere in the deep past, simple cells learned to communicate, to divide labor, to trust one another. And from that trust, every complex organism on Earth was born. The unseen majority. When we think of life, we often think of the visible. Trees, animals, people, oceans full of motion. But by sheer number, cells of our own kind are a minority. The planet is dominated by the microscopic, bacteria and other single-celled organisms. Outnumber multicellular creatures by a staggering margin. In a single gram of soil, there can be more living cells than there are humans on Earth. In the oceans, in the clouds, even in the rocks deep below the surface, microbes thrive. An invisible network of life that sustains the planet's chemistry. They recycle nutrients, generate oxygen, and form the foundation of every food chain. It is estimated that over 90% of Earth's total biomass exists in the form of single-celled life. We are, quite literally, inhabitants of a microbial world, a shared heritage. Despite their differences in form and function, all cells share a common ancestry. Every living thing on Earth, from the smallest bacterium to the tallest tree, descended from a single ancestral cell that lived more than 3.5 billion years ago. This cell, the ancestor of all life, carried within it the essential machinery that we still see today: a protective membrane, a genetic code, a way to harness energy through countless generations. That original pattern was copied and modified, branching into the immense diversity of life we now know. In a sense, every living organism is a different answer to the same ancient question: how can matter come alive? The mathematics of life. When scientists try to describe this microscopic world, they often turn to numbers to quantify the unthinkable. A single milliliter of seawater can contain 1 million bacteria and 10 million viruses. Your own body hosts as many microbial cells as human cells, a partnership known as the microbiome. Together, these organisms influence digestion, immunity, even mood and behavior. You are not just one organism, but an ecosystem, a living landscape of interdependent cells and species. Every breath, every heartbeat, depends on these unseen collaborations. In every direction we look, life reveals itself in scale. From the forests that stretch across continents, to the microorganisms drifting unseen in a single drop of water. And at the heart of all of it, the cell. A structure so small it escapes the naked eye, yet so numerous that together they form the living world. To understand the cell is not only to see its design, but to grasp its scale, its size, its number, and the astonishing diversity that makes life possible. The numbers within us. The human body contains an estimated 37 trillion cells. It is a number almost beyond imagination. If you were to count one cell every second, it would take you more than 1 million years to finish. Each cell functions as a miniature factory producing energy, processing information, and communicating with its neighbors. Yet no single cell perceives the whole. Together, they build the human being. Even this enormous total is only part of the story. Alongside our own cells live billions of bacteria microbes that form the body's microbiome. On the skin, in the mouth, and deep within the intestines, they exist in mutual partnership, outnumbering the stars in the Milky Way many times over. To be human, then, is to be a host, a walking ecosystem of cells. Some belonging to us, some not, but all sharing the same space. Measuring the invisible. When Robert Hooke first peered through his microscope in 1665, the cell was a mysterious pattern of empty boxes. Today, we can measure those boxes with astonishing precision. A typical human cell spans 10 to 30 micrometers in diameter, about 1/10 the width of a single strand of hair. To place that in perspective, a sheet of paper is roughly 100,000 micrometers thick. thousands of cells could fit along its edge. The scale grows smaller still. Within the cell are organelles, some only a few hundred nanometers across. DNA coils into strands just 2 nanometers wide, so thin that 50 million of them could lie side by side across a fingernail. And beneath that, the atomic realm. Proteins folding, ions moving, electrons exchanging energy. A cascade of scale, from the visible to the unimaginable. Across the vastness of Earth's living world, life presents itself in endless forms: towering forests, soaring birds, hidden microbes. Yet, beneath all this variety, every creature is built upon the same foundation, the cell. It is difficult to grasp the enormity of that fact. For the cell, though invisible to the naked eye, is the most abundant living structure on our planet. In its numbers, in its diversity, and in its intricacy. It defines what life is and how it endures. A hidden multitude. In the body of an average adult human, there are estimated to be 37 trillion cells. Each one alive, each one carrying out its role in quiet precision. It is a number so immense that the mind struggles to imagine it. If you were to count every cell in your body, one per second, you would not finish in a lifetime. Not even in 1,000 lifetimes. And yet, those countless cells are you. Your bones, your blood, your skin, your brain. Every tissue, every organ, every thought that flickers through your mind arises from their silent cooperation. Cells die and are replaced constantly. Every second, millions complete their life's work and vanish, while millions more divide and take their place. It is a cycle of renewal that never stops. A rhythm as old as life itself. The smallest of worlds. To understand the scale of a cell, we must journey into the microscopic. A typical human cell measures between 10 and 30 micrometers across. A micrometer is 1/1,000,000th of a meter. A scale far beyond what our eyes can perceive. To put it in perspective, a single grain of sand is about 1 millimeter wide. You could line up roughly 100 human cells across that distance. And yet, inside each of those microscopic spaces exists an entire functioning system. A living world enclosed within a boundary, complete with power plants, transport networks, and communication systems. Within those few micrometers, a cell generates energy, constructs proteins, reads genetic code, and makes decisions that sustain the life of the organism. It is, quite literally, a universe within a speck. The largest of cells. While most cells are impossibly small, a few can be seen without magnification. The human egg cell, or ovum, is the largest cell in the human body, measuring roughly 1/10 of a millimeter across. It is barely visible to the naked eye, a tiny sphere of potential from which a human being can grow. Beyond the human body, some cells are vast by comparison. The yolk of a bird's egg, for example, is a single giant cell filled with nutrients to sustain the developing embryo. In the oceans, certain algae produce cells that stretch several centimeters across. And among the most remarkable of all is the single-celled organism called Lerpataxifolia, a green alga that can grow to the length of a human arm. Its entire body composed of one continuous cell. At both extremes, microscopic and macroscopic, cells show a flexibility that defines life's resilience. On the opposite end of the scale lie the bacteria and archaea, the simplest and smallest of living cells. Some are only 0.2 of a micrometer across, so small that 50 of them could sit side by side, across the width of a single human hair. Yet even at this minute size, these cells contain everything necessary for life: a membrane, genetic material, and the molecular tools for metabolism and reproduction. They live in every environment imaginable, from boiling volcanic springs to the frozen polar ice, from the ocean depths to the thin air of the upper atmosphere. They thrive within our bodies, and beneath the soil beneath our feet. The sheer number of microbial cells on Earth defies comprehension. It is estimated that there are 5 nonillion bacterial cells on our planet. That is, 5 followed by 30 zeros. They outweigh all animals combined, and form the unseen foundation of the biosphere. Every leaf, every drop of water, every grain of soil teems with microbial life, the unseen engines of our planet's ecosystems. A diversity of form and function. When we look closer, we see that cells are not identical copies of one another. They are as varied as the roles they perform. In the human body alone, scientists recognize more than 200 distinct types of cells. Each one carries the same DNA, yet expresses different portions of it to become specialized. Muscle cells form long fibers that contract and generate movement. Nerve cells stretch out like living cables, carrying electrical signals across great distances. Blood cells circulate through the body, delivering oxygen and removing waste. Some cells spend their lives dividing rapidly, replacing tissues that wear away. Others, like neurons, may last a lifetime, preserving the continuity of memory and experience. In plants, diversity is equally striking. Photosynthetic cells capture sunlight, transforming it into chemical energy. Root cells absorb water and minerals. Guard cells open and close tiny pores, controlling the exchange of gases. Everywhere, form reflects function. The architecture of the cell is shaped by the task it was designed to perform. The architecture of cooperation. While single-celled organisms can live independently, most life as we know it is multicellular, built from vast communities of cells working together. This cooperation allows for specialization and complexity. When cells unite, they form tissues. Tissues organize into organs, and organs into systems, each contributing to the functioning of the whole. Within this hierarchy, no cell operates in isolation. Chemical signals flow continuously between them, coordinating activity and maintaining balance. In a human body, cells form networks as intricate as any city. They exchange nutrients, transmit information, and repair one another when damage occurs. Through this constant communication, the body maintains its delicate equilibrium, an internal harmony that sustains life. Every living thing, from the smallest bacterium to the vast complexity of the human body, depends upon a boundary. Without it, there would be no separation between order and chaos, between the living and the non-living. That boundary is the cell membrane, a structure so thin it cannot be seen even with a light microscope. Yet it defines the very existence of life itself. It is the line that marks where a cell begins and where it ends. It is the frontier between the self and the world beyond, a delicate wall between worlds. The cell membrane, sometimes called the plasma membrane, is astonishingly thin, only a few nanometers thick. To imagine its scale, consider this. If the wall of a single cell were magnified to the thickness of a sheet of paper, the cell itself would be the size of a house. Yet this microscopic barrier is both strong and flexible, delicate and dynamic. It is the barrier that keeps the chemistry of life contained. Without it, the molecules that make up living matter would simply dissolve into the surrounding environment, and life would cease to exist. The membrane's brilliance lies in its simplicity, a design that has endured for billions of years, unchanged across nearly every organism on Earth. The architecture of the membrane. At the heart of this structure is a double layer of lipid molecules, the phospholipid bilayer. Each phospholipid molecule has two parts: a head that is attracted to water, and a tail that repels it. When placed in water, these molecules arrange themselves naturally, the heads facing outward toward the water, the tails tucked inward, away from it. This self-assembling sheet forms a barrier that is stable, flexible, and perfectly suited to life. The outer surface of the membrane faces the watery environment of the cell's surroundings. The inner surface faces the equally watery interior of the cytoplasm. Between them lies a narrow region of oily tails, a zone that repels water and prevents most substances from passing freely through. This elegant arrangement is not fixed. It flows, bends, and moves. A fluid mosaic, as scientists describe it. Proteins drift within it like ships on a calm sea, carrying out the work of transport, communication, and recognition. The term fluid mosaic model captures both the order and the flexibility of the membrane. Fluid, because the lipids and proteins within it can move laterally, sliding past one another with ease. And mosaic, because it is a patchwork of many components: lipids, proteins, and carbohydrates, each with its own role to play. This fluidity is essential. It allows cells to change shape, to divide, to repair damage, and to interact with their environment. If the membrane were rigid, life would be static. But because it is dynamic, life is possible. Selective permeability, the gatekeeper of life. The most remarkable feature of the cell membrane is its ability to control what enters and what leaves. It is selectively permeable, not a solid wall, but a carefully regulated gateway. Some substances, like oxygen and carbon dioxide, slip easily through the lipid layer. Others, like ions or large molecules, require assistance to cross. Embedded within the membrane are proteins that serve as channels and carriers. They act as gates, pumps, and sensors, moving materials in and out. and ensuring that balance is maintained. Through these proteins, nutrients enter, wastes are removed, and signals pass between cells. This continuous exchange keeps the internal environment of the cell stable and distinct, a state known as homeostasis. In this way, the membrane is not merely a barrier. It is an active participant in the cell's survival, a living interface between the organism and its world. The language of signals. The cell membrane is also a communication device. Scattered across its surface are proteins and carbohydrates that act as molecular identifiers, tiny markers that allow cells to recognize one another. When hormones or neurotransmitters reach a cell, they bind to these receptors on the membrane's surface. The binding triggers a cascade of events inside the cell, like a key turning a lock. Through these signaling systems, cells can sense their surroundings, respond to environmental changes, and coordinate with their neighbors. In a human body, billions of signals are sent across membranes every second. Each heartbeat, each thought, depends on the precision of this communication. The membrane is the antenna and the messenger, listening and responding, ensuring that the orchestra of the body plays in harmony. The cell membrane must strike a perfect balance. It must protect, but it must also connect. A membrane that sealed the cell too tightly would cut it off from the resources it needs to survive. A membrane that was too porous would dissolve the distinctions that define life. Through its selective permeability, the cell finds harmony between isolation and openness. It guards its interior, yet allows the exchange of matter, energy, and information. This balance is not static. It shifts continually, adjusting to changes in temperature, pH, and pressure. Even in the harshest conditions, deep sea vents, polar ice, desert heat, cells maintain this equilibrium through subtle adjustments to the composition of their membranes. Fatty acids may change shape. Proteins may reorganize. The chemistry of the boundary adapts to preserve life within. It is resilience at the molecular level, a triumph of adaptation written into every cell. The art of control. The cell's ability to regulate its internal environment depends entirely on the membrane's precision. Ions such as sodium, potassium, calcium, and chloride must be maintained in exact concentrations. Transport proteins act like tiny pumps, using energy in the form of ATP to move these ions against their natural gradients. This process creates electrical potential, the voltage difference that powers nerve impulses and muscle contraction. Without the disciplined control of the membrane, the body's most basic functions would fail in an instant. The quiet work of countless microscopic gates keeps the heart beating, the mind thinking, and the body alive. Membranes and energy. Beyond transport and communication, the cell membrane also plays a vital role in energy production. In bacteria, the membrane is the very site of cellular respiration, where electrons are passed through protein complexes, creating gradients that drive the synthesis of ATP. in the mitochondria of eukaryotic cells. A similar process occurs on the inner membrane, folded into structures called cristae. Across these membranes, energy flows like an electrical current, powering every reaction that sustains life. Membranes, therefore, are not just boundaries. They are engines. They are the sites where energy and matter meet, where the chemistry of life is transformed into motion and growth. The continuity of a design. Despite the immense diversity of life, the design of the cell membrane has changed little over billions of years. Whether in a human neuron or a bacterial microbe, the same phospholipid bilayer performs the same essential tasks. Its composition may vary. Different lipids, different proteins. But its function remains universal. This continuity is evidence of nature's efficiency. When evolution finds a solution that works, it endures. The membrane's form is not merely convenient. It is inevitable. A structure perfectly tuned to the needs of life. If the cell membrane is the boundary of life, then what lies within is its world. A landscape invisible to the naked eye, but vast in its complexity and quiet activity. Inside every cell, beyond the delicate wall of the membrane, is a shimmering, translucent space. It is not empty. It is not still. It is filled with substance, a gel-like matrix known as the cytoplasm. And within it, a hidden architecture of filaments and fibers. The cytoskeleton. Together, these form the stage and the scaffolding of life. The medium in which every process unfolds. The sea within. Imagine shrinking yourself down to the scale of a molecule, small enough to drift through a single living cell. At that level, the interior would not appear as a void, but as a thick, glowing sea, a suspension of countless molecules, each in constant, purposeful motion. This is the cytoplasm, a substance neither fully liquid nor solid, a colloid of water, salts, proteins, and nutrients. It makes up the majority of the cell's volume. In a human cell, it occupies the space between the nucleus and the membrane, embracing every organelle in a soft, dynamic embrace. If one could listen to it, the cytoplasm would hum with energy, the faint vibration of molecules colliding, reacting, exchanging. It is here that most of the cell's chemistry occurs. Here that enzymes catalyze reactions, that sugars are broken down, that proteins are folded and transported. It is not merely a background. It is an ocean of activity, where every wave and current sustains the rhythm of life. The composition of the cytoplasm. The cytoplasm is composed primarily of water. But this water is no ordinary fluid. It is crowded with solutes and macromolecules, so densely packed that a single drop contains more order than any engineered machine. Floating within it are ions such as potassium, sodium, calcium, and chloride, each crucial to maintaining electrical balance. Enzymes drift freely, catalyzing reactions in microseconds. Lipids form temporary droplets. Sugars dissolve and reappear as energy flows through. And suspended throughout are the organelles, the nucleus, mitochondria, endoplasmic reticulum, and many more, each held in place, yet subtly swaying, in the currents of the cytoplasmic sea. Scientists often distinguish between two regions here: the cytosol, which is the fluid portion, and the cytoplasm, which includes everything within the cell, except the nucleus. Together, they create a living medium, a material that supports structure while enabling motion. In many types of cells, the cytoplasm does not sit still. It flows through a process known as cytoplasmic streaming. Materials are moved from one region of the cell to another. Nutrients, organelles, and chemical signals swept along invisible pathways. In plant cells, these flows are particularly striking, circling the large central vacuole in gentle currents that can be observed under a microscope. The flow ensures that even the most distant parts of a cell receive what they need: oxygen, glucose, and messages from the nucleus. Without it, the cell would stagnate. With it, life moves effortlessly within the cytoskeleton, the cell's hidden framework. But beneath the apparent fluidity of the cytoplasm lies a hidden order, a framework of structure and strength, known as the cytoskeleton. It is invisible to the naked eye, but if we could illuminate it, the interior of the cell would appear like a vast network of filaments and bridges, stretching in all directions. This network gives the cell its shape, its ability to move, and its internal organization. It is the scaffolding that holds life together from within. Three filaments of form and motion. The cytoskeleton is composed of three main types of protein filaments, each with its own character and role: microfilaments, intermediate filaments, and microtubules. Microfilaments, the fine threads. At the smallest scale are the microfilaments, also known as actin filaments. They are thin, flexible threads made of the protein actin, arranged in twisted chains that line the inner surface of the cell membrane. Though only about 7 nanometers thick, these filaments form dense networks that give the cell its shape and tension. They are responsible for the cell's ability to move and contract. In muscle cells, actin interacts with another protein, myosin, to generate the forces that cause contraction, the very movement that drives every heartbeat, every step, every breath. In other cells, actin filaments allow motion on a smaller scale, the crawling of white blood cells toward an infection, the graceful changes of shape that occur during cell division. They are the filaments of motion, the muscles of the microscopic world. Next are the intermediate filaments, thicker than actin but thinner than microtubules. They are composed of various proteins, depending on the cell type: keratin in skin cells, vimentin in connective tissues, neurofilaments in nerve cells. These filaments provide tensile strength, resisting stretching and mechanical stress. They form a supportive mesh that holds the cell together, anchoring organelles in place and connecting them to the membrane. In a sense, they are the cell's internal cables, absorbing tension and preventing rupture. Without them, cells would tear under their own movements. With them, they remain resilient, flexible, but unbroken. Microtubules, the highways of the cell. At the largest scale are the microtubules, hollow cylinders made of the protein tubulin. These filaments are the cell's highways, rigid, dynamic structures that extend outward from a central hub, called the centrosome, near the nucleus. Microtubules serve as tracks, along which vesicles, organelles, and even entire chromosomes are transported. Tiny motor proteins chitosan and dynein walk along these tracks, carrying their cargo like microscopic delivery workers. With every step, they convert chemical energy into motion, moving essential materials through the dense cytoplasm with astonishing precision. During cell division, microtubules form the mitotic spindle, a structure that aligns and separates chromosomes, so that each new cell inherits a complete set of DNA. In certain cells, microtubules also form structures such as cilia and flagella, whip-like appendages that allow movement and sensing. It is through the graceful rotation of flagella that bacteria swim, and through the synchronized beating of cilia that our airways clear dust and debris. Without microtubules, the organized traffic of the cell would grind to a halt. Dynamic instability, the living scaffold. Unlike the bones of an animal, the cytoskeleton is not fixed. It is constantly assembling and disassembling, adapting to the cell's needs in real time. Microtubules grow and shrink. Actin filaments polymerize and dissolve. Intermediate filaments rearrange as the cell changes shape. This phenomenon, called dynamic instability, allows the cytoskeleton to be both stable and responsive. When a cell migrates, filaments extend in the direction of motion. When it divides, they reorganize to form the mitotic spindle. When it rests, they reinforce its shape. The cytoskeleton is not a static frame, but a living, breathing architecture. Deep within the quiet interior of every living cell, past the shifting currents of cytoplasm and the delicate framework of the cytoskeleton, there lies a singular and vital chamber. It is large by cellular standards, spherical and luminous beneath the microscope's gaze. This is the nucleus, the guardian of the cell's most precious possession, its genetic code. Everything that the cell is, everything it will ever become, is written and stored here, within this silent library of life. The command center. The nucleus serves as both archive and control center. It contains nearly all of the cell's DNA, a molecule that carries the complete set of instructions for building, maintaining, and reproducing life. Every protein, every enzyme, every structure the cell produces is directed by the information encoded within these strands. Through a process of exquisite precision, the nucleus decides which instructions to follow and when. It is the cell's administrative core, a place of regulation, coordination, and memory. Without it, the cell would lose its identity, its order, and its future. The nuclear envelope, a fortress with gateways. The nucleus is enclosed by a double membrane, known as the nuclear envelope. This barrier separates the genetic material from the rest of the cell, protecting it from harm, while maintaining communication with the cytoplasm beyond. Each layer of the envelope is a lipid bilayer, similar in composition to the cell membrane itself. Between the two membranes is a narrow space called the perinuclear cisterna, through which molecules can move and interact. But the nucleus cannot be sealed entirely. Information must flow in and out. Instructions must leave. Materials must return. This exchange is made possible by nuclear pores, complex gateways that pierce the envelope, like tiny portals between two worlds. Each pore is a vast structure, built from hundreds of proteins, and capable of regulating what passes through. Small molecules diffuse freely, but larger cargo such as RNA or proteins require permission, escorted by transport factors that recognize and guide them safely across. Through these openings, the business of life continues. Messages depart, materials arrive, and balance is maintained between inside and out. The nuclear envelope is, in this sense, both fortress and bridge, a structure that protects without isolating, that connects while it defends. Within the envelope lies the nuclear plasm, a clear, viscous fluid that supports the contents of the nucleus. It serves as the medium in which the genetic material, the chromatin, floats and functions. The nuclear plasm contains ions, enzymes, and nucleotides, all of which contribute to the continuous maintenance of DNA and RNA. Here, in this transparent environment, the most delicate molecular machinery operates. To the human eye, the interior of the nucleus may seem uniform and still, but at the molecular level, it is a place of constant activity, a microscopic library in perpetual motion, its volumes being read, copied, and rewritten at every moment. Chromatin, the threads of information. The nucleus's central treasure is chromatin, a mixture of DNA and proteins. If the DNA from a single human cell was stretched end to end, it would measure nearly 2 meters in length. And yet it fits within a nucleus only a few micrometers wide. This remarkable feat of organization is made possible by the way DNA is packaged. The molecule is wound around proteins called histones, forming bead-like structures known as nucleosomes. These coils fold further, looping and condensing, until the DNA is compact enough to fit neatly within the nucleus, yet still accessible when needed. Chromatin exists in 2 main forms. Euchromatin, which is loosely packed and active, where genes are read and expressed, and heterochromatin, which is tightly coiled and silent, a vault for unused or temporarily repressed genes. Through this elegant architecture, the cell can access only the information it requires, when it requires it, preserving order amid unimaginable complexity. The nucleus, the factory within. Nestled within the nucleus is a darker, denser region called the nucleus. It is not separated by a membrane. but its presence is unmistakable. A concentrated hub of activity, visible even under a simple microscope. Here, one of the cell's most vital processes unfolds: the production of ribosomes. Ribosomes are tiny molecular machines that assemble proteins from amino acids, and nearly every function of life depends upon them. Within the nucleus, the genes that encode ribosomal RNA are transcribed, and these RNA molecules are then combined with proteins imported from the cytoplasm. The result: pre-ribosomal particles, which are exported back through the nuclear pores to complete their assembly in the cytoplasm. From this quiet chamber, the tools of life's construction are continually produced and sent forth, a steady supply chain feeding the cell's endless needs. The nucleus is, in essence, a factory within the library, an engine built at the very heart of the blueprint itself. The DNA within the nucleus is a molecule of staggering significance. It carries the complete instructions for the structure and function of every cell. This information is written in a 4-letter code: adenine, thymine, cytosine, and guanine. The sequence of these bases forms the words and sentences of the genetic language. Segments of DNA that contain specific instructions are called genes. Each gene provides the code for making a particular protein, the molecules that form the body's tissues and drive its chemistry. When a cell needs to produce a protein, it does not remove the DNA itself. Instead, it creates a copy, a messenger RNA molecule, which carries the information outward. This process, known as transcription, takes place within the nucleus. Enzymes such as RNA polymerase bind to the DNA, unwind its double helix, and assemble a complementary strand of RNA based on the genetic code. Once the message is complete, the mRNA exits through a nuclear pore and travels to the cytoplasm, where ribosomes translate it into a protein. Through this elegant choreography, the nucleus governs all that the cell does, issuing instructions, receiving feedback, and maintaining the continuity of life. Chromosomes, the library's bound. When a cell prepares to divide, the chromatin fibers condense into distinct structures called chromosomes. Each chromosome is a single, continuous molecule of DNA, coiled and compacted to ensure its safe transfer to the next generation of cells. In humans, there are 46 chromosomes, arranged in 23 pairs. One set comes from the mother, and one from the father, carrying within them the combined legacy of ancestry and evolution. As the cell divides, the chromosomes are duplicated and separated, so that each new cell inherits a full set of genetic instructions. It is an act of perfect precision, a molecular copying that has occurred billions of times, with astonishing fidelity, since the dawn of life. In the vast stillness of night, as your body rests and the world outside grows quiet, an unseen world continues to stir within you. Trillions of cells, each one alive, each one working in perfect, silent coordination. They repair what has been worn. They build, replace, and renew. They listen for signals, share messages, and sustain the life that is you. Every thought that drifts through your resting mind, every slow, steady breath, is powered by this endless, microscopic symphony. Within their tiny boundaries, molecules move with purpose. Energy is created and shared. Information is copied and passed on, as it has been since the first spark of life appeared on Earth. The same chemistry that moves within you tonight once moved through ancient seas, in the earliest cells that began the story of all living things. And so, in your quiet rest, you are connected, not only to every other person, but to every creature that has ever lived. All part of the same delicate lineage, the same flowing current of life, written in the same universal code. So as you drift towards sleep, know that the work continues. Peaceful, patient, tireless. Inside you, the universe hums softly on, cell by cell, breath by breath. And tomorrow, when you open your eyes once more, that same quiet universe will awaken with you, ready to begin again. The mind of a plant does not live in a single place. There is no brain. Instead, every part of the plant thinks. The roots decide where to grow. The leaves decide when to open. The flowers decide when to bloom. It is a distributed intelligence, a harmony of many small choices, woven together into a single life. Perhaps that is why plants seem so peaceful. They do not rush to conclusions. They simply respond, adjust, and endure. In a forest after the rain, the air smells alive, earthy, rich, full of unseen messages. That scent, called geosmin, is produced by microbes in the soil. Plants use it, along with other chemical signals, to understand what is happening around them. Rain, decay, warmth. Each one tells a story. Plants listen, not with ears, but with their entire being. There are even plants that remember the length of days, the chill of winter, and the return of light. This memory allows them to bloom at just the right time each year. A tulip never guesses. A cherry blossom never rushes. They know. They measure the world in patience. Each day, each night, each degree of temperature. Slowly, steadily, they prepare. When conditions are perfect, they awaken as if on cue, unfolding in silence, right on time. Plants teach us that awareness does not need speed. It does not need language. It only needs attention. They notice what changes, adapt to what comes, and remember what has passed. And in their stillness, there is wisdom, the kind that watches, listens, and waits. Perhaps intelligence is not measured by how fast one can think, but by how deeply one can respond. Now imagine yourself as a leaf, spread open under soft light. You can feel the air around you, the temperature, the moisture, the faint pull of gravity below. You sense the presence of others, roots nearby, branches above. You do not think in words. You simply feel. The sun warms you, and you take that warmth in, turning it to energy, to life. This is awareness. Simple. Complete. The intelligence of plants is not the same as ours. It does not rush to understand. It simply lives in perfect harmony with the world around it. It is a kind of wisdom that we, too, once knew. The knowledge of rhythm, of season, of balance. When you walk through a garden or a forest, you are walking among quiet thinkers, patient beings that have spent millions of years learning how to live well, not through control, but through cooperation, not through speed, but through stillness. So tonight, as you rest, let that stillness reach you. Breathe in and imagine the wisdom of the forest flowing through you like sunlight through leaves. You don't need to solve anything. You don't need to hurry anywhere. Like the plants, you already know how to exist in balance. You already know how to listen to the world. And when you wake, the same intelligence that guides them, that quiet awareness, will still be there within you. The story continues, leaf by leaf, breath by breath, root by root. The slow, living intelligence of the green world never stops. It only grows. And so, as our story comes to its quiet close, the world of plants continues. Softly, patiently, endlessly. All around you, life is growing. In the cracks of the pavement, in the soil beneath your feet, in the forests that breathe on the edge of cities, and in the gardens that wait for morning. Every root, every leaf, every seed moves in rhythm with the same calm heartbeat of the earth. They are the gentle builders of our world, turning sunlight into air, air into breath, and breath into life. And tonight, as you rest, you are part of that same quiet circle. The oxygen in your lungs was once a leaf's gift. The food that nourishes you was once sunlight stored in green. The calm that fills you now is the same peace that moves through every living thing. Take one deep, easy breath and imagine the sound of leaves stirring in the night air, the faint rustle of grass, the whisper of trees. This is the lullaby of the planet, a music that never ends. The plants will keep breathing, growing, turning toward the light, even as you drift away into sleep. So let your thoughts settle like seeds in soft soil. Let your body rest like a tree in winter. Let your mind open like a flower at dawn. You don't need to do anything more tonight. The story continues. In the roots, in the leaves, in you. Good night. And may you dream in green.